Hello. Today I'll be discussing the critical role that pollen nutrition, specifically protein content and source diversity, plays in the health of the western honeybee, Apis mellifera, at both the individual bee and colony level. Let's start by introducing our subject, the western honeybee, known scientifically as the Apis mellifera. Why is this insect so important? Apis mellifera is a critical animal pollinator for a vast number of our food crops and wild plants, making it essential for both global food security and healthy ecosystems. They also provide valuable products like honey and wax. But beyond its ecological role, Apis mellifera is a key model organism in science. Its complex social structure, communication methods, and colony organization make it extremely valuable for research into social behavior and general insect physiology. So what fuels these colonies? Honeybees rely on specific resources gathered from their environments. Nectar, collected from flowers, is the main source of carbohydrates, providing the energy needed for flight and other activities. It's also converted into honey for long-term storage. Pollen, however, is uniquely important as it's their sole natural source of proteins, fats, vitamins, and minerals. These nutrients are absolutely vital for raising young bees, for the proper development of adult workers, and ultimately for the health and productivity of the entire colony. Water is also essential for various physiological and hive maintenance needs. This presentation will focus specifically on the critical role of pollen nutrition. Now, a key challenge regarding pollen is that its nutritional value isn't uniform. Pollen from different plant species can vary dramatically in protein content, from 1.5% to 48.4%, and also in the lipid content, from 1.2% to 24.6%. Furthermore, the surrounding landscape plays a huge role. Agricultural areas dominated by single crops offer very limited pollen diversity compared to natural meadows and forests. This variability leads to the central hypothesis guiding much research, that both the sheer amount of protein bees get and the diversity of pollen sources they access are critical factors for their overall health and ability to cope with stress. How does the protein concentration and diversity of pollen sources impact key individual and colony health indicators in Apis mellifera? These indicators can range from individual bee development and immune function to colony-level factors like pathogen resistance and overall growth. Our first finding relates pollen quality to individual bee development, specifically focusing on the hypopharyngeal glands, or HPGs, which nurse bees use to produce essential brood food. This figure from DePasquale and colleagues shows the size of the HPG acini, a measure of gland development, in bees fed different single-source pollens and mixed or no pollen. The key findings from this figure are clear. Bees fed no pollen had underdeveloped glands. Most importantly, the quality of the monofloral pollen significantly impacted development. Bees consuming rubus pollen, which the study identified as highest in protein and antioxidants, developed significantly larger gland acini compared to bees fed lower quality cistus or erica pollens. This demonstrates that higher pollen quality directly fuels better development of this vital physiological trait. Interestingly, the mixed pollen diet resulted in development similar to the average across the individual pollens tested. Moving beyond single sources, does pollen diversity impact immunity? This figure from Alot and colleagues examines glucose oxidase, or GOX, activity, an enzyme contributing to social immunity by helping sterilize food within the colony. The key findings from this figure are striking. First, consuming any pollen boosted GOX activity compared to bees fed no pollen. More importantly, both polyfloral blend diets resulted in significantly higher GOX activities compared to monofloral diets, even when those single pollens had the same or higher protein levels. Furthermore, there were no significant differences in GOX activity among the different monofloral pollens themselves, irrespective of their protein content. This strongly suggests that the diversity offered by a polyfloral diet, rather than just the protein quantity of a single source, specifically enhances this aspect of social immunity. Now, how does nutrition affect resilience when bees face direct challenges like pathogens? The study by DePasquale specifically investigated how different pollen diets affect the survival of bees infected with Nosema ceranae, a widespread and damaging gut parasite implicated in honeybee health declines. This figure shows the survival curve specifically for those Nosema-infected bees. The key findings are evident. Bees fed the nutritionally poorest pollen, cistus, died the most rapidly after infection. In contrast, bees fed with the highest quality monofloral pollen tested, rubus, survived significantly longer. Interestingly, the polyfloral mix provided a survival benefit equivalent or near equivalent to the best single source pollen, rubus, and significantly better than the cistus erica or castanea diets. This highlights that dietary diversity can be particularly beneficial under infection stress. Furthermore, the study found that longer survival in these infected bees correlated positively with higher baseline activity in the immune enzyme phenyloxidase, suggesting that diets supporting better immune function contribute directly to enhanced pathogen tolerance. In conclusion, the research highlights that pollen nutrition is multifaceted and vital for honeybee health. We've seen evidence that pollen quality, influenced by nutrient content, is fundamental for physiological development, like the hypopharyngeal glands. Secondly, pollen diversity offers distinct advantages over single sources by enhancing aspects of social immunity, like GOX activity. Thirdly, good overall nutrition achieved through access to both high quality and diverse pollen resources directly bolsters bees' abilities to tolerate pathogen infections like nosema. Furthermore, it's important to note that the connection between diet and the bee's internal ecosystem, the gut microbiome. The type of pollen consumed influences the microbial community, which in turn significantly impacts the bee's own metabolism, weight gain, and overall health state, adding another layer to why nutrition is so crucial. Taken together, both the nutritional quality and diversity of pollen are crucial factors driving Apis mellifera health, impacting their physiology, immune responses, pathogen resilience, and interactions within their essential gut microbes. The significance of this research extends beyond the hive. For beekeepers, it emphasizes the importance of managing colony nutrition through ensuring access to good, diverse, natural forage or using high-quality supplements when needed. For agriculture and land use, it underscores the potential detrimental effects of large-scale monocultures that limit pollen diversity and the benefits of practices that promote diverse floral resources. Fundamentally, understanding these nutritional requirements is vital for conserving honeybees and the crucial pollination services that they provide worldwide. Future research needs to continue exploring these complex interactions, particularly how nutrition intertwines with other stressors like pesticides and pathogens, and further identify key micronutrients or pollen components vital for bee health. Thank you so much for listening. This is the listed 5 peer-reviewed references for you.